So I'm just going to start the recording now. So yeah, so just to start off, so hello everyone, thanks again for logging in today to the latest in the MSc webinar series. My name is Paul, and today we're going to discuss multicolor flow cytometry panel design. All right, my slides aren't advancing. Okay. So before I start, I'd just like to introduce you to the MSc Life Science team. So my name is Paul, as I said. I've been with MSc for 2.5 years now. Before I worked with MSc, I did my PhD and a postdoc in NUI Galway. And among the techniques where I have a lot of experience are flow cytometry, cell culture, and ELISAs. Lisa is as the life science sales manager. So Lisa got her PhD from the University of Leeds, and she has 7 years postdoc experience. And her expertise is in molecular biology, protein expression, and Western blotting. And then Emma joined MSc about a year ago now, and after 2.5 years as a postdoc, and she has experience in cyto and histochemistry, flow cytometry, and Western blotting. So just a little bit of an agenda for today. So we will focus today on designing a multicolor flow cytometry panel, and I've broken up the talk into a few different areas. Now, they do all link together because they're all obviously very related. So we'll talk about knowing your cytometer, knowing the properties of your fluorophores, knowing the important things to know about your cell population. Then we'll look at designing the panel itself. And finally, we'll look at intracellular staining and a few other factors, and a few other factors that are important. Sorry, I don't know what I did there. In your flow cytometry multicolor panel. Okay, so this presentation, I think some of you were at the first sort of flow cytometry webinar we did last year. So it's a follow-up to the one that we did last year. That was an introduction to flow cytometry. It was a broader introduction to the technique itself. The video is on YouTube or on the MSc website if you'd like to refresh your memory on some of the more basic principles or techniques. And if anyone can't find that, just let us know. We can send you a link. So this is a slide from that previous webinar, but I think it's good to pop it up here again so we can ask ourselves and refresh ourselves on what exactly flow cytometry is. So flow cytometry is a technology which can simultaneously measure multiple physical characteristics of single particles. So these physical characteristics can be around the size of a particle, the particle's granularity, fluorescence emitted by the particle, and the particles themselves then can be a range of different things, including cells. They can be beads, whether those beads are fluorescent or not, or they can be other small things like small multicellular organisms, generally in the range of kind of 0.2 to 150 microns. So when designing your panel, it's really important first that you know about your cytometer and the inner workings of what you've got in there and what you don't have. So each cytometer is made up of a series of different parts. So you've got your excitation lasers, you have lenses and prisms for focusing the light, there's a flow cell, you'll have dichroic mirrors, and you'll have bandpass filters. You'll also then have a series of photomultiplier tubes, which collect the light and turn it into electrical signals that your software can understand. So each of these pieces play a really important role in your panel design, and it's important that you know and understand what they do and the impact that this might have on the choices available to you in terms of fluorophores later on. So the first part, I'm just going to go into a little bit of detail on some of the particularly important bits in terms of multicolor panel design. So the first part of the cytometer are the lasers. So as we all know, a laser is a light source which emits a very specific wavelength of light. And this light in the cytometer is directed towards your cells and is used then to excite your fluorophores. So I've picked out a few of the more common lasers that you might find on your own cytometer. And as you can see, they vary through a wide range of the light spectrum from UV down here at short wavelengths, 355 nanometers, all the way up to far red lasers, around 750 nanometers. So the more lasers your cytometer has, the more, in theory, fluorophores you can use, and the more flexibility you're going to have when you're looking to design your panel. The next part of the cytometer, which you need to take into account when you're choosing fluorophores are the dichroic mirrors, or as sometimes we call dichroic filters. So these come in two types. You'll have short pass or SP, and you'll have long pass or LP. And they'll always be denoted like this, a number followed by SP or LP. So what that number means, so basically the number, if it's a short pass filter, that it will let all wavelengths below that number through, or in the case of a long pass filter, it will let all wavelengths above that number through. So here we can see in this 575 long pass filter that it's letting all wavelengths of light greater than 575 nanometers through, but it's reflecting away the light below 575 nanometers. Now, in most cytometers, the inner workings aren't linear, so that doesn't necessarily mean that this light is discarded or it doesn't go in the bin or anything. It can go through another filter, go down a different path. So there are complex and almost sometimes 3D things to take into account here. The next part then is bandpass filters. These are generally the last filter that the light would pass through before it reaches your PMT and is converted to electrical signals. So the nomenclature again of these is a wavelength, which is the first number. So in this case, 630 nanometers, slash another number. In this case here, we have 20. So we have a 630/20 filter here. So that means that the wavelength allowed through by this filter is 630 plus or minus 10 nanometers. So 20 nanometers around the headline wavelength. So 630 and 20 around that. So plus or minus 10. So usually a cytometer will have bandpass filters installed which correspond to the peak emission of like kind of common fluorophores. So for example, a very common one that you'll see is 530/30. This can often be called kind of the FITSI filter. So these filters will let in a short bandwidth of light and they'll reflect everything else back. So this then goes on to the photomultiplier tree generally. So next thing now is to talk about knowing the different characteristics of the fluorophores that you're going to encounter and that you might use in a flow cytometry multicolor experiment. So there are hundreds, if not thousands, of different fluorescent molecules which can be used in flow cytometry. So these include single dyes, such as PE, FITSI, APC, PERCP. These have been around for many years, but then there are also slightly newer dyes like the Alexafluors or the brilliant violet dyes. And these have been specifically developed to have brighter fluorescence and more photostability. And the newer dyes have also been developed to take advantage of more lasers that have come on the market, like the 355 and the 405 lasers. And also then in the case of the brilliant violets particularly, they have increased the Stokes shift, or the difference between the excitation and the emission, to give you more possibility for multiplexing. So these are very common. You'll see them all over the place. The next type are tandem dyes, and these are the second major type of fluorophore. And these comprise a small fluorophore here, coupled to another one. So you've got PE coupled to Cy7. So with the first dye, in this case PE, is excited, it transfers its energy to the second dye, or the acceptor, in this case Cy7. So this activates the Cy7, and this Cy7 then produces a fluorescence emission. This process is known as Forster resonance energy transfer, or FRET, and it's a useful way to produce a higher Stokes shift and therefore increase the number of colors that can be analyzed from a single laser. So there are a wide variety of tandem dyes available. For example, here we see PE Cy7. You get examples of PERCP Cy 5.5, PE Texas Red, APC Cy 5.5. There's lots of different ones available. The next major type of fluorescent molecules that you can analyze using a cytometer are fluorescent proteins, widely used in biology, such as like GFP, RFP, M-Cherry, and they're very commonly used as reporters in cells for reporting the expression of a particular gene, or as fusion proteins. And they often allow you to detect an intracellular marker without having to permeabilize the cells. This can be particularly useful for some experiments around sorting, where you might want to get live cells back, but to sort based on the expression of some intracellular protein. And then, like any technology, flow cytometry is advancing and changing and evolving all the time. And there are new dyes coming along along. So one of these new dyes are the Caravia dyes from Sony. So these dyes incorporate an organic backbone, which separates then a number of different fluorophore molecules. And this minimizes quenching effects. As well as minimizing the quenching, as you can see here, it also allows more different fluorophore molecules to be tagged to the same antibody. So this allows a relatively dim fluorophore like FITSI to actually be very bright because you're just multiplying the number of FITSIs that you've got in there. And this can be really useful for detecting very weakly expressed or very rare markers, like a very rare cell surface receptor that might be changed by a small amount on a cell after treatment, or maybe an intracellular transcription factor or something like that. So these are really cool, and they're kind of a new technology that's coming along, developed by Sony. And then we won't really go into it today, but there are newer techniques altogether, such as mass cytometry. So these don't use fluorescence, and instead the antibodies are tagged with metal atoms. And this combines flow cytometry with mass spec to determine the levels of antibody binding. And like you can achieve massive multiplexing here. You can go to hundreds of different antibodies because of the wide variety of metal ions and metal atoms available. So again, with a fluorophore, they all work in a similar way. So they all work by fluorescence. So it's really important to understand the fluorescence characteristics of fluorophores. So I'm sure we all know, but we'll run over it again, that fluorophores are excited by one wavelength of light. This results in an electron taking up the energy from the light and moving from a ground energy state to a higher or excited state. The electron, though, is unstable at this higher state, and it can't stay here for long. So it emits this extra energy that it has picked up as light, so it can move back to its ground state again. The light emitted then in a fluorescent molecule is at a higher wavelength than the light which excited it. For example, here you can see that we excited this electron at 488 nanometers, but it emitted light at 507. This is really useful because this is what we can detect. So we're able to then distinguish between these two different lights and pick up this one. And we know what's fluorescent. So every fluorophore then will have an excitation and emission spectral profile. So here we can see just a generic example of one. So every fluorophore has one of these, and it's definitely worth keeping the spectras for the fluorophores that you use handy when you're designing your panel. And we'll see why and how useful it can be to know this later on. So as you can see, there's a range over which the fluorophore is excited, which is the first curve here on the left. This is called the absorption or the excitation curve. And the emission wavelength then is then shown as a separate curve. So this is the wavelength of light that's emitted after it's as fluorescence. So here we can see that it's a higher wavelength than the absorption spectrum, which you can clearly see. And the difference between the peak of the absorbance and the peak of the emission is what's known as the Stokes shift. You do see often some spectral overlap between the excitation and emission. This obviously means that you may get some excited, like the light that's used to excite the fluorophore coming into the emission profile when you pick it up, but it's just something to be aware of. It can always be compensated away at the end. But the best fluorophores generally will have a high Stokes shift, so a long distance between the excitation and emission. So it's really easy to distinguish between the light used to excite the fluorophore and the light emitted because of fluorescence. Another thing to remember is that not all fluorophores are the same. So they're not all created equal. What do we mean by this? So if we think about fluorescence and the physics of how it works, so every fluorophore is a different chemical makeup. So each fluorophore absorbs different levels of energy at different wavelengths. And because of this, they emit different amounts of photons, different intensity of light, at also at different wavelengths. So all of this taken together means that fluorophores have different relative brightness levels. So they're not all as bright as each other. And there's some handy tools online. For example, the BioLegend website has its fluorophore brightness index. And this lists many of the fluorophores, not all, but there are 50 to 60 fluorophores. They're all the common ones. And their relative brightness levels from 1, which is the dimmest all the way up to 5, which are the brightest. So I've just picked out a few of the common fluorophores here with their relative brightnesses just to show you. So you can see fluorophores like PE, PE Sci 5, and even like APC here and some of the brilliant violet dyes. They're really quite bright. But common fluorophores that you might use every day, like FITC and PERC-P, are relatively dim in comparison. So, you know, PE has a brightness level of 5, FITC only has a brightness level of 1. So, does this mean that, you know, you should always try to use fluorophores with brightness levels of 4 and 5 so that everything's really bright and, you know, it's great? Kind of not really. We will see in a little while where it's advantageous to keep the relative brightness levels of your fluorophores in mind when you're going to design your panel. And you can actually take advantage of these features to make your analysis more straightforward, more robust as you go through. So we'll just keep that in mind and we'll come back to it later. The next thing that's quite important, if you can, is to know the characteristics of your cell population before you start with a complex multicolor panel. So some of the important things to note are the relative expression levels of the markers you're interested in. So I've made this this really biologically accurate cell here. So this is our cell of interest, and we can see that we've got 3 markers on the surface that we're interested in. So we've got red receptor, we've got a green receptor, and we've got a gold receptor. So as we can see, there are different numbers of each of these antigens or each of these receptors on our cell surface here. We've got 6 of the red ones, we've got 3 green ones, but we've only got 1 gold one. So this is the same with cells. So if you're looking at different markers on cell surface, one of them could be very abundant, while others might be very, very rare. So it is quite useful to have this knowledge or have as much of it as you can before you start jumping into an 8 or a 10 color panel. But where can you find it out? So, like, is there somewhere where all this is listed? So that there are in the literature, you'll be able to find some information on this. There are also some online resources. For example, the BioLegend website has this expression of common surface molecules on blood cells table, which goes through all the common types of white blood cells and a lot of the more common markers on them, and gives you the number of estimated number of molecules per cells. Also gives you references, which is quite useful. You may be looking at a marker that's not listed. You could find this out yourself, so you can get some antibodies for the markers you're interested in, all of them with the same fluorophore. Stain them all, run them all, and see which one is the brightest. That could be, if you want to try and do that, the one with the highest relative fluorescence is the most abundant, the lowest is the least abundant. So that's important to know, and again, we'll see how that fits in with the brightness levels later on. The next thing to think about is dreaded autofluorescence. So basically all cells have some level of autofluorescence, and it can be caused by the presence of collagen, elastin, cyclic ring compounds like NADPH, riboflavin can cause it, so can organelles like mitochondria or lysosomes. Few things to note about autofluorescence. You'll often see it at lower wavelengths, for example, in the FITC channel. So this can cause problems when you're trying to analyze your results. It can reduce the sensitivity and it can give you false positives if it's particularly bad. So having good controls can definitely help. But if you have very autofluorescent cells, sometimes if you know this, you can use different fluorophores and avoid the channel altogether, thereby minimizing the problem. So it does help to know if your cells are very autofluorescent before you start. Another thing to think about, and it's not just, doesn't really just go for big multiplex panels, it really across all flow cytometry, is that your staining process isn't killing an undue amount of your cells. So if you're killing a really high proportion of your cells before you start analyzing them, you can run into big problems. You can lose your data completely. You can get a lot of autofluorescence. So, runs into a lot of problems. So you really want to check that your staining protocol is staining all your cells, but not killing them all as well at the same time. So it's a good idea always to have a cell viability dye in your experiments so that you can exclude the dead cells at the beginning of your gating strategy. This is becoming easier and easier recently. You can get fixable viability dyes now, like Ghost dye from Tombow, which make this much easier and more flexible. And they come in all the different colors as well, so you can fit them into any panel really. The next thing is antibody titration, which I know when I was in the lab wasn't something I was ever excited me too much or anything. It is a slightly labor-intensive process. It can be a bit of a pain sometimes, but it can actually be a really important part of optimizing your panel and ensuring that your results are nice and robust. Especially true if you're looking to identify a rare population or you're looking at fine changes in expression levels of particular markers. It takes time, but it can also save you time. It can save you money, and it can make your data better. So it can save you a bit of heartbreak, can save you having to go back to repeat an experiment or something. So why would we titrate our antibodies? Well, you improve your staining accuracy first of all. So we can see here this is a titration experiment where we've got different concentrations of antibodies going from right to the highest on the right to the lowest on the left. And as you can see, really high concentrations. This potentially negative or very weak population is actually getting more and more intense with the increased concentrations. So a well-titrated antibody, you want to see the maximum separation between your positive and negative populations in your sample. So if you've got too much antibody, you can end up with increased levels of non-specific binding. This increases the spread of your negative cells like we see here. And it can result in suboptimal staining and an inferior separation of your positives and your negatives. So, ideally here you'd possibly go with something like this, which you're seeing a good robust separation between positive and negative, but you're not seeing any reduction in the positive staining, which you're starting to see at this concentration here. So another thing, as I said a minute ago, titrating your antibody will often save you money. So in my experience, and I'm sure in most of yours as well, you'll often find that you end up using less antibody than the recommended manufacturers, that the manufacturer recommends, sorry. So therefore you'll stretch your vial of antibody a little bit further. This of course isn't great news for the likes of me. I'll end up selling you less antibodies, but there we go. So just a few tips for titrating your antibodies. So you should always use the same staining conditions that you're going to use in the real experiment. You should try to test a range of different antibody concentrations, both above and below the manufacturer's recommendation, just to be sure. And you should always acquire the same number of live single cells per tube. This goes, of course, across the board, like for titration experiments and your analysis experiments. You should always be analyzing the same comparable numbers. So this is just a little run through a gating strategy for your cells here for looking at in titration. It's an example. I couldn't find a full one with that ended up like this. But what you do, you select your cells, gating out all the crud, which would be here on the left, and cell debris and things like that. You then want to gauge your single cells. So you want to do side scatter area on the X versus side scatter height. Get your nice single cells, gating out all your doublets and triplets and things. In here, we've got a viability dye. So they've actually used a ghost dye here, and they're gating on their marker of interest, which is an IgD marker. But we've gated out all our dead cells. So we've gotten rid of those from our analysis. And then we're here able to look and distinguish nicely our positive and negative. So that's what you want to see. That's a well-titrated antibody. There's good definition there between your positive and your negative population. So now that we've kind of know about our fluorophores, we have looked at the expression levels of the markers on our cells, we've titrated our antibodies, we can then move on to maybe thinking about putting a multicolor panel together. So when I was putting this presentation together, I was trying to put these things in order so we could kind of go in sequence from one thing to the next. But that doesn't really make sense because you have to think about them all together. So I'm going to kind of go through them all together, and we'll bring things in and out as we go, and hopefully it'll all make sense. But we'll talk about things like spectral overlap, relative marker expression level and why that's important. Then we'll move on to some of the tips and tricks and things to think about if you've got intracellular markers in your multicolor panel. Linked to this is fixation of cells, and then some other things just to be aware of at the end. So we'll bring back in our cell here, and we'll consider what we know now. So we know we want to detect these 3 markers on the cell surface, And this goes if you've got 3 markers or you've got 10 markers. The same principle applies. I've just done 3 today because it's, just for clarity. So if we look at our red receptor, it's very abundant. We've loads of copies of it. So we want to pick a dim fluorophore here. Our green receptor, we have kind of a medium level of expression. There's 3 of them, not loads, but not very rare either. So we're going to pick maybe a medium fluorophore in terms of brightness intensity. But then we've got our gold receptor, which is very, very rare, very few copies of that per cell. So we're going to pick a nice bright fluorophore to make sure that we pick up any of our gold receptor that might be there, or pick up any differences between our treatment groups. So we know what fluorophores we should use, what types of fluorophores we should use for each of our markers. But the question now is which one to pick. So, okay, we don't know. So if we look very simply at our needs on the left, we need a dim one, we need a medium one, and we need a bright one. So we might as well just jump in straight away and say, okay, we need a bright one, we'll use PE. We need a medium one. All right, well, what's that? 2, 3, or 4? We'll pick PE size 7 here. And we need a dim one for this red receptor. So all right, we'll pick FITSI. Okay, so we've picked a medium one, a bright one, and a dim one. So here we go. So it seems like a reasonable choice. We have a nice bright fluorophore for our gold receptor in PE. We have a medium one for our green receptor in PE size 7. And we have our dim one for a really abundant receptor in FITSI. Now, another advantage we have after picking these 3 fluorophores is that these are 3 pretty common fluorophores that that a large number of different suppliers are going to have in their catalog. So we'll have a lot of choice. It's likely that, antibodies tagged with these, because they're very common, they're going to be the cheaper of the antibodies. So let's just take a look at how this panel might go on the cytometry. So here we can see the emission the excitation spectrum of these 3 fluorophores. So I have put FITSI here in red because that's for the red receptor. I've put PE size 7 in green because it's for the green receptor. And I put PE in gold because it's for the gold receptor. So we can see the 3 excitation profiles here. And as you can see, it's quite a lot of overlap between them. Now, because these are excitation profiles, this in itself isn't a major issue because we're not detecting the light that's exciting them. But, it's just to be aware of it. On most cytometers, just to note, these 3 fluorophores will be excited by the same laser, which is shown here in the blue. So they'll be excited by the blue laser, which is at 488 nanometers. So as we say, not a big deal, just something to be aware of. So the next question then to ask is, what does the emission spectra of these three fluorophores look like? And what impact does this have on our data analysis later on? So I've done that here. So here are the emission spectra of these 3 fluorophores. And straight away, we can see that we will definitely have to do some compensating here. So again, we've got our FITSI in red, which we can see here with a peak emission of around 525. PE in the gold, kind of browny gold color, peak emission around 574. And then you've got PE size 7, whose peak emission is over here, the 7, I think it's the 770s. So if we take FITSI and PE first, we can see the FITSI's emission peak is around 525. These peaks are quite well separated, the peak emissions. So it's certainly possible to run these 2 fluorophores together in the same panel, but you can see that there's actually quite a bit of overlap in the emission spectras of FITSI and PE. So therefore, some of the signal picked up by the detector for PE will actually be coming from FITSI, and vice versa in this case. So this, of course, can be compensated out, but you really need to know this before you start. You have to be alert to this. Now, there isn't too much of a problem if we look with FITSI and PE size 7. As you can see, they're really far away on the peaks, are really far away on the spectra. There is a small peak here for PE size 7 around the PE area. There's going to be no FITSI spillover into the PE size 7 channel, which will be detected up here around 780. But you can see there may be a little bit of spillover from the PE size 7 into the FITSI. So again, you need to be aware of that. You need to be aware that you might have to compensate. So that it shouldn't be a major issue. There is then going to be, of course, some significant enough spillover from PE size 7 into the PE channel. As you can see, it's right at the peak of it there. So again, you know, compensation here, your controls are really important. That's going to take you time. You know, you're going to want to be sure that you're doing it right. So you need a bit of experience. So, you know, maybe is there a better way to go about this? Is there better options we could pick? So if we go back here and we'll look at our requirements again. So as we said, we need a dim one, dim fluorophore, we need a medium one, and we need a bright one. But now we're really thinking about how we might separate the emission spectra of our fluorophores as much as we possibly can, while staying with the relative brightness levels that we've already identified that we need. So I did a bit of background reading, and we could go with these choices. So now we have, instead of FITSI for our very abundant red receptor, we've used Pacific blue, which has the same relative brightness level. For our medium fluorophore, instead of using PE size 7, I've chosen Brilliant Violet 711, which again has the same relative brightness. And we've left PE as our gold receptor. So now we should see no difference in the intensity of the staining. But what about the separation of our emission spectra? So we can now see that we've a really good separation in the emission spectra of each of our markers. So if you ran these as single stains on your cytometer, you'd see that there's very, very little spillover from Pacific blue here in red into the PE channel, and vice versa from PE into Pacific blue. And this is going to make your compensation much, much easier, much more straightforward, and give you a lot more confidence in your results. It'll make your analysis easier, and you're far less likely to run into issues during your analysis with some spillover or something like that, or running into a problem because you didn't compensate them properly. So your data is going to be much more robust, and you can have a lot more confidence in it. And again, it's just an easier experiment to run. So we'll pop back to our checklist for designing a panel. So we've looked at spectral overlap, and we've kind of highlighted the importance of that. In tandem, we've looked at the relative marker expression level, and we've kind of gone through why that's important, or where you'd use that. But there are some other things that you want to take into account when you're designing a multicolor panel. So for example, are you doing any intracellular staining? And if you are, you're definitely fixing your cells. So these things go hand in hand. But are you fixing your cells as well? You know, for if you've got a huge amount of samples and you want to run them, you can't run them all in one day. So there's loads of reasons why you'd fix cells. So some things to take into account in terms of this. So a good way to talk about fixing an intracellular staining is for us to look at how we would stain an intracellular protein and integrate this into our panel. So again, we'll go back to our same cell again. So we have the red, green, and gold receptors on the cell surface in the same quantities. But we also now want to stain this intracellular protein here in the kind of dark gray, black intracellular protein. This could be a lot of different things. It could be a cytokine, it could be a transcription factor, it could be any other intracellular protein. Indeed, there are ways that you can detect mRNAs now by flow cytometry using oligos, which are tagged with fluorophores. And this can add a whole other layer to a multicolor panel. So it doesn't really matter what it is, but we'll just say today it's a protein just for simplicity's sake. So I'm just, again, in our previous webinar, there is a bit more time spent on how you stain intracellular proteins. But I'll just really quickly run over this just for anyone who hasn't seen that webinar or mightn't be overly familiar with intracellular staining. So the first thing you need to do is you need to stain all of your cell surface markers first in the normal way. So we see that here where we've got all our antibodies and they've come in and they've bound to our cell surface molecules. At this point as well, if you want to use a fixable viability dye, this is the time to do that. Next, then you need to permeabilize your cells. So this is represented here with these pores that I've generated. Often you will fix your cells first with PFA or an alcohol-based fixative and then use a permeabilization reagent. Your antibodies then for the intracellular protein are added in the permeabilization reagent, and they are allowed to enter the cytoplasm through these pores which are made, and they can bind then to your protein of interest inside the cell, which we can see here. You then wash off, all the excess antibodies, excess reagents, and you're ready to go on your cytometer. So this is what you'll have going to your cytometer. You're going to have a cell with antibodies bound to all your surface markers and your intracellular markers. So if we go back to our cell and our fluorophore brightness index, and we'll try and pick out some fluorophores for our now 4-color panel. But is there anything we need to take into account in particular because our 4th protein is now an intracellular protein and because we're fixing our cells? So the answer is yes. So there are a couple of things we do need to take into account. So one of the major obvious differences between staining intracellular markers and cell surface markers is obviously that you need to fix and permeabilize the cells. So there are a few things to take into account. The first is you need to make sure your fixation doesn't alter the epitopes of your proteins too much. Nowadays, this isn't a major problem because there are some really good, really well optimized reagents from various different suppliers out there. Like Tombow and BioLegend, well, others are available. And by following the recommended manufacturer's protocols, you'll often be fine here, but it is worth making sure. Next thing is to make sure that you are taking care when using tandem dyes. So we mentioned tandem dyes at the very beginning, for example, PE-Cy7 or APC-Cy 5.5. They're really useful for expanding the number of markers that you can use. But you need to be careful because the acceptor dye, like the Cy7 or the Cy 5.5, can end up being knocked off with hard treatment, such as fixation. So while it's definitely possible to use tandem dyes on fixed cells or with intracellular staining, you do need to make sure that your protocol is okay with this, that it's not damaging the dyes, that you're not seeing a reduction of intensity or funny, that you're getting the acceptor knocked off. So you're ending up with staining, for example, in the PE channel or something like that. There are more and more alternatives now to tandem dyes that in the same range. For example, you could substitute APC Cy 5.5 out and put in Lexafluor 700, which has very similar fluorescent profile. So that's something to just be aware of. And just like rare cell surface markers, you should choose a bright fluorophore for intracellular staining. There are a few reasons for this. If you think about it, the intracellular environment is quite busy. There are other proteins in there. There's other fluorescent proteins. There's mRNA. There's DNA if you're staining a nuclear protein. And these could all interfere with the emission of the light. So the light from your fluorophore, as well as that, has to travel through more liquid. It has to go through the cytoplasm. It has to go through the lipids of the cell membrane. All of these things can absorb light and refract it off in different directions. So you need to have a brighter marker to help overcome this. So now we'll take these guidelines into account and let's pick some fluorophores for our panel. So we'll just go back here and the only change we've added is now for our intracellular protein, we need another bright fluorophore. So we need a dim one, a medium one, and 2 bright ones. So our old panel that we used for our cell surface proteins used PE, Brilliant Violet, 711, and Pacific Blue. Now, happily, all of these are actually fine because their brightness levels are okay, their emission profiles are okay, and also none of them are tandem dyes. So we're perfectly happy to keep those. So let's keep them. And we can add in something like APC for our intracellular fluorophore. So APC will give us what we need. So we'll have it's quite a bright fluorophore. So it's 4 on the brightness scale. So it'll give us a good strong intensity. And there's very little spectral spillover into our other channels, as you can see here. So now we've got our 4 fluorophores on the spectral profile here, the emission. So we've got Pacific Blue, PE in gold, APC fits in here in purple, and our Brilliant Violet 711 over here. So we do obviously have a little bit more compensation potentially to do here, but it's, the peaks are relatively well separated. It should be doable. That is going to happen the more fluorophores you add. You're not going to be able to avoid some level of spillover, but that's a pretty good panel. It gives us everything we need. So just some other factors before we finish up that you need to think about when designing a multicolor panel. So we've covered spectral overlap, we've looked at relative brightness, we've looked at intracellular staining in a bit of detail, but then there are other things. So controls. So first off, every flow experiment should have unstained cells. These are used for setting your cell size and your granularity gates and for defining your negative populations. You should always then have single cell controls or single stain controls, I apologize. So this is cells stained with just one of your each different type of antibody. You need these for compensation. Whether your spectral overlap is limited or not, you still should make sure that you're compensated correctly. The final type of control then you need are FMOs, or fluorescence minus ones. So you need these for drawing your gates, for defining your positive populations. You can, of course, use them just antibodies, so leave out one of your antibodies, or you can substitute an antibody for an isotype control. Either way is fine. You should also think about the potential for non-specific binding. This is particularly important if you're looking at phagocytic cells like macrophages, because the macrophages have Fc receptors on their surface. These will bind non-specifically to antibodies. And because the cytometer can't tell the difference between a specifically and a non-specifically bound antibody, this can cause a serious issue. So you should consider whether you need to do an Fc block on your cells before you start staining. So just to recap really quickly, so to design a multicolor panel, it's important that you know the features of your cytometer, what channels you can use, what fluorophores you can use. It's then important to know about as much as you can about those fluorophores, their intensity levels, their emission spectra, what type of fluorophore they are. All of these things are important. You should know about as much as you can about your cells. The more you know, the easier it is to design an effective panel. Titration of your antibodies is important and should be done. You should always use brighter fluorochromes for rare markers, and vice versa. You should use dimmer fluorophores for very abundant markers. Again, similarly for intracellular markers, generally good to use a brighter fluorophore. You should be careful using tandem dyes with fixatives. Not saying it can't be done, just to check that your protocol isn't damaging your tandem dye, just something to be aware of. Controls, always really important. And like anything with flow cytometry or any other real technique in biology, to be honest, to have your technique optimized is really important. The more optimized it is, the more robust the technique is, the better your data out the end is going to be. So that's key. So yeah, I'd just like to thank everybody for attending today. It's great to see a good turnout. I hope you enjoyed it. I hope you learned something from it. To thank you, we are going to send you a discount code for a discount on any of our flow cytometry products. So we'll be sending that out to you in the next, probably tomorrow. And you can get in touch with any of us. So Lisa, Emma, or myself. Our email addresses are here. You can get in touch if you've got any questions or concerns, or you want to order anything. We're always here to help. And I'd be very happy to take any questions that anyone has. I'll just close this down and see if there are questions in the Q&A. And thanks for listening.
Thanks very much, Paul. Yeah, there's a little Q&A button at the bottom if anyone wants to use it. I don't know if you can unmute people.
Yeah,
Paul, if you want to.
I think.
Cool, you might unmute everyone, which might freak them out.
Yeah, I think people can unmute themselves. Oh, I have an allow to talk button. So if I'll just click allow to talk on everybody, and then if you want to speak, you can unmute yourself. Or you can, if you don't want to talk out loud, feel free to type anything into the Q&A bar. Sorry, apologies, Ty. I asked you to unmute and I didn't mean to. Yeah, so. Okay, so no questions. Okay, that's good. Yeah, so look, thanks again to everybody. I hope you got something out of it. And yeah, as I say, we're always here for questions or concerns, or if you need more information, or you need, quotes or whatever. And don't hesitate to contact us. So thanks again, and we hope to see you at the next webinar in a few weeks' time. So yeah, have a nice day, everyone. Bye bye.
Thanks, Paul.